Okay, Allan, well, thank you for joining us. And as you know, we've been following your company a lot over the years, and congratulations on your progress. It's one of the very interesting success stories of merging two very exciting technologies, cell therapy and oncolytic viruses. Maybe we can begin by telling us a little bit about the history of the company.
Well, the history of the company, the co-founder, Dr. Salai, he founded Genelux in 2001. It was a naked adenovirus, virus Lister strain. And he learned the first wave of oncolytic virus looked good on paper, it looked good in mouse studies, but he realized it went to the human body, the whole dynamics changed with the complement, and 9 to 10 pints of blood is different than a mouse getting an oncolytic virus. So when he founded Calidi Biotherapeutics, co-founded with me, he came up with the idea with stem cells of delivering the vaccine virus. And probably with Dr. Karen Abbvie at City of Hope, they were talking about it while he was in the previous company, Genelux. They didn't act on it until he left Genelux. So we've done a lot of research in the last 7 years, potentiating the virus. We know the optimum virus cell ratio and when to cryopreserve the product. So we've done a lot of research. Our scientists have been really good, and we're proud of them.
Let me interrupt you for a quick second, because I did not realize that. So you can actually infect the stem cells with the virus and then freeze the stem cells and then thaw them out, and the virus is still alive in the stem cell?
Absolutely. We've seen that when we cryopreserve it and we go to room temperature, and you inject it intratumoral, the virus is still expanding. So you have a factor, it's like a mini bioreactor. So you have expansion before you cryopreserve at an optimum peak of virus production, and then the ratio, and then you take the room temperature, you inject it in intratumoral, and there's also virus expanding to kill tumor cells. So that's the beauty of our product. It's the X factor exists throughout the continuum until it likes tumor cells.
So let me ask you, using your expertise in the area of stem cells, you've actually leveraged that as well in the area of COVID, is that correct?
Yes, the allogeneic cell bank that we harvested stem cells like 4 years ago from healthy donors through strict FDA guidelines. You have to be under 40, for example. You have to have no history of cancer and no history of this and that. And then we test the blood, and then we studied. Our scientists studied the cells and how they expanded, and so we picked this particular cell line and we GMP manufactured it. FDA approved it for COVID patients. We licensed that to Sorrento, and they actually did a phase 1 trial at UCSF, and they were all on ventilators. And then several weeks later, they got off ventilators and totally healed. So it reduces inflammation of the lungs. So there's a lot of products with our adipose allogeneic cells. We call it ethical cells, because it's not controversial. It's not umbilical cord, or it's not anything. It's not placenta. It's not. So it's the Vatican likes it because it's ethical, and it's just as powerful as bone marrow. It's reproducible. We can produce trillions of cells. Our German facility learned how to, with a bioreactor that we have, can really produce a tremendous amount, and without having to deal with any limitations.
No, it's a very, very exciting area. I don't know if you see it. Several weeks ago, there was a publication of a patient who received iPSC cells and ended up developing a teratoma. There are similar publications with fetal stem cells. So the danger in the stem cells is obviously in the fetal and embryonic is not just ethical, but it's also the risk of developing cancer. As you probably know, if you put embryonic stem cell in a mouse, it's an embryonic stem cell because it causes teratoma. Let us take a step back with the technology. Let's talk about yourself for a second. So maybe you can tell us about your background, because from what I recall, your background is quite a diverse background. It wasn't in biotech.
Yeah, I think what I bring is really the business strategy and acumen, and I have my software technology really automated clinical trials. So Johnson and Johnson used my technology. LabCorp used it. It's a patient engagement software and patient consent, HIPAA compliant, 29C Part 11. So all the data can be accessed through mobile devices. You can consent, you can have adverse reporting, you can have images of the melanoma or the tumor size going down and share it with the physicians. So there's a lot of data and engagement that's dynamic. We can also get.
When did you develop that?
I started developing in 2010 area, and we sold the company in 2017.
Congratulations.
And I'm using that technology in Calidi. So I'm part a little bit of business ability to do deals. We did 7-figure deals with J&J and PPD, which is a CRO, and also a deal with PRA Health, which got bought by Icon. So we're actually doing early licensing strategies with our assets early. We have a lot of traction, so we'll probably generate revenue sooner than most biotechs would do.
Yeah.
Have a strong business team along with a good scientific team.
So what would you advise to somebody who wants to follow a similar career path to you? What are some of the things to do? What are some of the things to avoid? Is an MBA very important? Just in general, what are your thoughts and advice to younger people?
Well, if you're a business person, MBA gives you the tools, but if you don't like business, you don't like the messiness of that, then an MBA won't do it because you really have to have a natural desire. People realize that being a CEO, you spend 50% of the time doing what you don't want to do. And so, they always say a startup, you have certain rights as a startup, a right to lose sleep, a right to get sued, a right not to get paid, a right for your spouse to be, mad at you, a right to lose sleep. I mean, there's a lot of rights, right? And starting up a company isn't what people, you have to rethink realistically why you want to do what you're going to do.
Okay. Well, Allan, thank you so much for your time. We really appreciate this. And periodically we will check in because I'm sure listeners are very curious on your progress.
Thank you. Thank you, Tom.
Thank you. Have a good weekend, Allan. Bye-bye.
Bye-bye.